Hello everyone, my name is Bertrand David, a senior researcher, and I am currently conducting my research in tissue engineering at CentraleSupélec, Université Paris Saclay in France. Without further ado, I will start my presentation, which serves as an introduction to life engineering. In this presentation, we will first define engineering and engineering for health, then we will explore the concepts of cell and tissue therapies through its two components, which are tissue engineering and cell therapy. We will then continue with a focus on stem cell science and briefly discuss the notion of organoids. Finally, as a conclusion, we'll discuss the applications of cell and tissue therapies. Let's start with engineering. So, what is engineering? Engineering, generally speaking, is a study that covers the technical, economic, financial, and social aspects of an industrial project while coordinating the work of several teams of specialists. And life engineering, particularly, is a field of engineering that is mainly concerned with health, agri-food, environment, and new energy. Still, you may still be wondering what is engineering for health. Health engineering integrates the principles of physics, chemistry, mathematics, computer science, and engineering to study biology, medicine, behavior, and health. The main goals are to advance fundamental concepts, deepen our knowledge on the molecular level as well as the organic level, and develop innovative biological products, materials, processes, implants, and devices. The main fields of application are risk prevention, medical diagnosis, treatment of pathologies, rehabilitation of patients on disability, and improvement of human health. But, as we all may know, no field exists without its challenges. In the case of engineering for health, researchers often struggle in understanding and describing the overall functions of living systems in their environment, regulation, mechanism, dysfunctions, evolutions, etc. And in modeling, designing, and mastering living systems and technologies in order to study, implement, enhance, or repair them. One essential characteristic of engineering for health is that it is a multidisciplinary activity that requires a pooling of skills in biology, process engineering, as well as mechanics and many more. In fact, engineering for health is organized around strongly intercorrelated themes such as in vivo diagnostic imaging, bioprocess engineering, computer science, micro and nanobiotechnologies, biomaterials, and cellular, tissular, and organic biomechanics. I will say a few words on each of these themes. First, biomechanics and health. The cellular, tissular, and organic biomechanics is based on mechanics of fluids and solids, biomaterials, and tissue engineering. Fields of application are medicine, biology, bioprocesses, imaging, and robotics. The objective is to develop new prostheses, artificial organs, processes to regenerate tissues, decision support for surgeon on pre- or post-operative assistance, and finally, assistance related to age or disability. Secondly, imaging and health. In vivo, imaging is a very wide field that goes from functional to interventional imaging. Physiopathology, that is to say the study of disorders of the functioning of the body or its parts during disease. Developmental biology is a science that investigates how a variety of interacting processes generate an organism's heterogeneous shapes, size, and structural features that arise on the trajectory from embryo to adult, or more generally throughout a life cycle. Cancerology, that is to say a branch of science that specializes in the study and treatment of cancer. And finally, preclinical and therapeutic pharmacology, that is to say the branch of medical sciences that studies the chemical properties of drugs and their classification. Third theme, micro-nanotechnologies and health. Again, micro-nanotechnologies and health is a very wide field, goes from the design of new devices for in vivo chemical and biological detection, diagnosis, therapeutic monitoring, or drug delivery. The optimization of the interface with living organisms, like medical robotics, neurosurgery, and finally to the design of new approaches to understand the functioning of living organisms. Let's move on to bioprocesses and health. Applications goes from the cells to the product to study synthesis, transformation, or degradation processes in agri-food, energy, green chemistry, or the environment. For health in particular, bioprocesses play a crucial role in the production of drugs, vaccines, and the development of artificial organs by tissue engineering. Now let's move on the links between computer science and health. Computer science allows the broadband data management in different research areas such as pharmacology, that is to study the epidemiology and evolution of diseases, for the study of pathogenic organisms and genetic disease. Imaging for the development of algorithms suitable for fast and efficient data acquisition or exploitation for non-invasive and mobile imaging. And finally, structural and systems biology to help understand the mechanism involved in complex pathologies and aid in diagnosis. Finally, robotics and health. The field of applications is divided into two groups: the assistance to the medicine doctor and the patient support. For the assistant to the doctor, we can see, for example, the robotic trajectory tracking in radiology and radiotherapy and the simulation of medical and surgical procedures. For the assistance to the patient, we can see, for example, the activation and functional stimulation of paralyzed limbs and the active prosthetics and orthotics. Now, for the second part of my presentation, I will be discussing cell and tissue therapies. Before approaching regenerative medicine, I will talk about the science behind cell and tissue therapies. What are we talking about? Through history, the cell has gradually been considered as a care tool. Cell therapy can improve our management of damages and dysfunctions in different parts of the body in order to repair, replace, and regenerate. About 400 years ago, Hippocrates postulated that the healing power of nature on the body contains within itself the power to rebalance the humors and to heal itself. He introduced the idea that it is possible to restore the functions of an injured part of the body through surgery. In 1993, Langer and Vacanti defined tissue engineering as an interdisciplinary field of applications bringing together principles of engineering and life science aimed to develop biological structures that are capable of restoring, maintaining, and improving tissue functions. The idea is to develop a functional and living biological substitute to counterbalance the loss of functionality due to age, congenital defects, disease, and non-functional organs. This leads us over time to the emergence of regenerative medicine as a field of biotechnology, the goal of which is to restore tissue and functional activities. Regenerative medicine aims to replace deficient cells or restore destroyed organs, which is why the development of regenerative medicine is based on the understanding of the biological processes involved in the normal functioning and the generation of tissues in embryology, organogenesis, cell signaling, growth factors, and stem cell biology. Regenerative medicine can be applied to two main types of disease, either degenerative diseases, for example Parkinson's disease, or pathologies involving the destruction of cells, tissues, or organs, for example diabetes. In order to achieve such applications, there are two action strategies. The first strategy is tissue engineering, consisting of the implantation of biological substitutes constructed in vitro, and the second is cell therapy, consisting of the transplantation of cells to grow new tissues. Cell therapy and tissue engineering are the main constituents of the regenerative medicine sector. Tissue engineering applies the principles of engineering and life sciences to develop biological substitutes that will restore, maintain, or improve tissue function. And cell therapy prevent or treat human pathologies by administering cells that have been selected, multiplied, and pharmacologically treated or modified outside the body. I'm going to dive deep now within these subfields. Let's start with tissue engineering. Tissue engineering aims to improve or replace biological functions through the combinations of cells, materials, and appropriate biochemical factors. Tissue engineering is an interdisciplinary field combining knowledge and processes from physics, cell biology, chemical engineering, and the science of matter. Typical treatments using tissue engineering are treated non-living tissue, for example, for example, heart valves, tissue from another part of the patient's own body or another patient's body on ex vivo organ reconstruction. As tissue engineering very often employs living cells as basic material, it overlaps with the applications of cell therapies. Tissue engineering covers 6 cross-cutting technological fields: biomaterials, cells, computer science, biomechanics, biomolecules, and tissue design. A few words on each of these 6 different technological fields. Biomaterials is a very important field of tissue engineering. Over time, there has been a transition from materials to biomaterials and finally to tissue engineering products. For example, before Christ, the use of silk suture to mend wounds of gladiators, or more recently the development of prostheses for amputees returning from the battlefields. Today, biomaterials are seen as scaffolding for cells. Biomaterials are used to direct the organization, growth, and differentiation of cells while providing physical support and chemical signaling. A scaffold must allow cell attachment and migration, deliver and retain cells and biochemical factors, enable diffusion of vital cell nutrients and expressed products, and finally exert certain mechanical and biological influences to modify cell behavior. Moreover, a biomaterial must have characteristics such as biocompatibility, that is to say, a general term describing the property of a material being compatible with living tissue. Biocompatible materials do not produce a toxic or immunological response when exposed to the body or bodily fluids. Non-immunogenicity, that is, immunogenicity is an immune response by an organism against a therapeutic antigen. Suitable resorption rates, and finally, an easy manufacture. A biomaterial can be natural or synthetic. Some examples of synthetic biomaterials: polylactic acid, polyglycolic acid, and coprolactone. And some examples of natural biomaterials: collagen, fibrin, alginate, hyaluronic acid, and extracellular matrix. Naturally, cells are a key player in tissue engineering. I will briefly discuss stem cells later in this presentation. Cells can be obtained from 4 different sources. From the same organism, autologous cells; from the same species, allogenic cells; from a different species, xenogenic cells; and from cell lines, immortalized cells. Biomechanics study the properties of the original tissues and identify the minimum properties required to ensure functionality. Today, it is well known that cells touch, smell, and then respond to the mechanical stresses of their microenvironment. This property of cells to respond to their mechanical environment is a key factor for bioconstruction. Mechanotransduction describes the ability of a cell to actively sense integrate and convert mechanical stimuli into biochemical signals that result in intracellular changes, such as ion concentration, activation of signaling pathway, and transcriptional regulation. Quickly, the last two technological fields. In tissue engineering, families of biological molecules play a crucial role, such as angiogenic factors to promote angiogenesis. Angiogenesis is a process of growing new blood vessels from pre-existing vessels. Growth factors, a group of proteins that stimulate the growth of specific tissues. Growth factors play an important role in promoting cellular differentiation and cell division. And differentiating factors involved in triggering cell differentiation include cell signaling, environmental influences, and the level of development of the organism. During cell differentiation in multicellular organisms, cells become specialized and take on roles such as those of nerve, muscle, and blood cells. To conclude this list, the last point is tissue design. Cell culture is an indispensable in vitro tool used to the development of tissue engineering. Two-dimensional cell cultures have been used for 100 years and are still a dominant method in many biological studies. However, 2D cell cultures poorly recapitulate the conditions in vivo. Recently, three-dimensional cell cultures devices like bioreactors have received remarkable attention in tissue engineering. Optimization of cell culture conditions is very critical in ensuring powerful experimental reproducibility, which may help to produce artificial organs. I will now address the second component of regenerative medicine, cell therapy. Cell therapy can be developed from functional differentiated cells or stem cells. The goal of cell therapy is to replace, repair, or increase the functions of damaged tissues or organs. The cells can come from the patient, a donor, another species, or a cell line. There are two main treatment strategies in cell therapy. Use of stem cells to replace the targeted cells, or use the healthy cells to produce a substance needed by the body. As they have the best capacities for differentiation and proliferation, iPS, adult, and embryonic stem cells are at the center of research and development for new treatments. There is a hierarchy for stem cells according to their differentiation abilities. For instance, pluripotent stem cells are cells that are capable of differentiating into all embryonic and extra-embryonic cell types. Pluripotent stem cells, embryonic stem cells, on the other hand, are capable of giving all cell types of an organism with the exception of extra-embryonic tissues. And finally, multipotent stem cells, adult stem cells, are capable of giving rise to many, but not all, cell types. In humans, it is estimated there are about 500 different types of tissue. Just an example of adult stem cells, mesenchymal stem cells, which depending on the signals they receive can differentiate into bone, cartilage, muscle, stroma cell, ligament and adipocyte or dermal tissue. Today, stem cells are a key player in tissue engineering. That's why I'm going to make a focus on stem cell science. First, the embryonic stem cells. Their ability to give rise to a wide range of cell types make embryonic stem cells a popular research tool for application in regenerative medicine. Embryonic stem cells are cell lines of embryonic origin in continuous renewal. Stem cell lines are derived from pluripotent cells of the inner cell mass of a mammalian embryo. Cells from the inner cell mass can come from embryos obtained by in vitro fertilization or by somatic cell nuclear transfer, also known as therapeutic cloning. In order to generate specific types of differentiated cells from embryonic stem cells, different parameters can be modified. The chemical composition of the cell medium, the surface of the cell culture dish, the insertion of a specific gene. Embryonic stem cells can remain undifferentiated for a long time provided that the culture is carried out under very specific conditions. The goal of current embryonic stem cell research is to develop reproducible and efficient protocols to guide the differentiation of embryonic stem cells to desired cell lines. Next, we'll move on to adult stem cells. In general, adult stem cells are rare and do not have the same developmental capacities as embryonic or fetal stem cells. Adult stem cells are partially differentiated progenitors on multipotent cells that can be found in adult tissues. There are about 20 main types of somatic stem cells in mammalians, each of which can generate cells of specific lineage, for example, liver, pancreas, bone, cartilage, etc. Progenitor cells are rather difficult to find and isolate. They share the two main characteristics of embryonic stem cells. They can multiply identically over a long period, long-term self-renewal, without showing any signs of aging. They can give rise to mature cell types, differentiated cells, which specialized morphologies and functions. With the right culture medium, multipotent progenitor cells can differentiate into a myriad of cell types without causing spontaneous tumors, unlike embryonic stem cells. Finally, induced pluripotent stem cells known by the abbreviation iPS. iPS cells are identical to embryonic stem cells, but they are obtained by genetic reprogramming of differentiated adult cells. These iPS cells can be obtained from healthy donors or from patients. In the second case, the objective is to faithfully reproduce the donor's disease to understand its mechanism and test therapeutic molecules. The reprogramming of differentiated cells into iPS cells consists of genetically modifying them to reactive genes and embryonic signals of the differentiation and unlimited proliferation characteristic of a pluripotent stem cell. Reactivating pluripotency turns off the differentiation genes expressed by the cell at the adult stage. Any proliferating adult cells can be used to generate iPS cells. Blood cells are the most used to their accessibility through blood testing. Nevertheless, other cells were easily reprogrammed. Skin fibroblasts, keratinocytes, adipocyte mesenchymal cells, epithelial cells, or even amniotic cells. The re-expression of 4 genes overexpressed in embryonic stem cells is sufficient and necessary to reprogram an adult cell. The c-MYC gene is particularly known for its ability to cause cells to proliferate. The actual reprogramming step takes about 2 weeks and has a very low yield. Only 0.5% to 1% of the treated cells become pluripotent again. There is therefore a resistance and natural obstacles to this reprogramming. The cells having received the reprogramming genes are placed in a culture medium favoring the maintenance of pluripotency. Those that regain their pluripotency will multiply and form clones. They are then amplified and tested for numerous criteria: absence of genetic and chromosomal abnormalities, presence of the various pluripotency markers. Ultimately, obtaining a fully qualified iPS line from adult cells takes about 6 months. iPS cells have the same advantage as human embryonic stem cells. They proliferate infinitely and can differentiate into all types of cells in the body. They can come from donors selected for their genetic heritage. This possibility allows the culturing of patient cells specifically to model their pathologies and study them. While embryonic cells can be easily accessible and are moderately controversial when used in research and therapeutic applications, using them for social purposes will cause an ethical uproar. Reprogramming cells will make the notion of infinite regeneration of tissues possible, creating what we call the prolonged man by modifying our dynamic with aging and death. Alternatively, it can lead to the acquisition of new physical abilities, leading to the rise of a new genre of market economies. Transhumanism, offering the solution to extend and increase life, would then become a social and anthropological reality that would redefine the question of time and power. Stem cells seem to be a powerful tool, but overall, some limitations have been identified in the development of cell therapies with stem cells, such as reproducibility of results in humans, because the studies are first done with animal models. The cost of clinical trials, several million euros, and the acceptance by medicine doctors and patients. The integration into medical practice may be long and difficult. The penultimate part of my presentation will focus on organoids. Organoids are a bridge between in vitro traditional cell culture flat flask and in vivo physiological three-dimensional environment in animal or humans. The idea is to propose more in vitro physiological models. This is based on the fact that embryonic development is a complex process that takes place in 3-4 dimensions. That many decisions regarding cell differentiation and the formation of tissue and organs, organogenesis, rely on the 3D architecture of the embryo, and that these phenomena cannot be reproduced in conventional monolayer cell cultures. Over time, the scientific community has proposed successive definitions of the word organoids. For example, in the 50s and 60s, the term organoids allude either to subcellular structures, tumors, or cellular aggregates. The definition varies according to the authors. More recently, the term organoid defined a 3D cell cluster derived from either primary tissue, embryonic stem cells, or iPS cells, capable of self-organization and self-renewal with similar functionality to the original tissue. Today, the term organoid means resembling an organ. Organoids are defined by 3 characteristics. The cells self-organize in vitro into a 3-dimensional structure characteristic of the organ in vivo. The resulting structure is made up of multiple cells present in that particular organ, and the cells perform at least some functions they normally perform in that body. Organoids are 3D structures derived from stem cells or progenitor cells that at a much smaller scale recreate important aspects of 3-dimensional anatomy and multicellular repertoire of their physiological counterparts and can recapitulate basic tissue functions. The main features are the concomitant presence of multiple cell types belonging to particular tissues, the general architecture of these drafts of tissues, and the capacity for self-organization, capacity of cellular system which originally shows no trace of organization of any kind, to rearrange itself spatially under the effect of autonomous mechanism emanating from the system itself, even if exposed to an homogeneous environment. In traditional models, cell composition is typically monotypic, while in organoid, multiplicity plus cell types coexist. Also, in traditional models, no too little original cellular architecture is observed, while an architecture close to that of the original tissue is observed in organoids. Finally, in traditional models, a limited ability to summarize tissue-specific function is observed, while tissue-specific functions are observed in organoids. Organoids seem to be very powerful tools, but overall have some limitations. It is indeed a question of manufacturing in vitro structures which mimic certain functions of the organs. All the team also point out that the absence of vascularization or innervation of these models produced in vitro is a limit that can pose serious problems. This illustrates the fact that organoids do not contain all the cell types of certain structures, blood vessels in particular, necessary for the functioning of an observable organ in situ, but only some of them. To conclude this presentation, I will briefly address the application of cell and tissue therapies. The maturity of the regenerative medicine market is low. The industrial profile is mainly composed of products in the early phase of clinical trials, phase 1 and 2. The total market covers the areas of skin, bone, cartilage, cardiovascular diseases, regeneration of or replacement of teeth and organs, etc. So many. It is likely that regenerative medicine will one day be used to treat a wide variety of pathologies. For example, current therapeutic applications are blood disease, bones, and cartilage regeneration. Therapeutic applications in development are heart and vascular disease, nephrosis, muscular dystrophy. And the potential therapeutic applications are neurodegenerative disease, bone and joint tissue disease, liver disease, and neuromuscular diseases. Thank you for your attention, and if you want, you can send me your question by email. My address is below. Thank you again for your attention.